Thank you, everybody, for coming, and thank you, Phil, for the introduction. So I'm going to tell you a little bit about microscopy and where microscopy has been and where I think it's hopefully going to be going. So the optical microscope's been around for 350 years, and for 250 of those years, it was really the dominant tool we had to try to understand the living systems below the level of the naked eye. Microscopes continued to increase in their power and abilities until at the end of the 19th century, a physicist, Ernst Abbe, realized that there was a fundamental limit to how small you could see with an optical microscope. And that limit is about 100 times bigger than the molecules that make up living matter. And so on the one hand, it's 100 times smaller than a cell, so we learned a lot, but we were stuck at being able to see at the most fundamental level what's happening inside of cells. And as a result, the microscope that you could buy in 1980 really wasn't all that different from the microscope that existed in 1880. There were better light sources and so forth, but the limits were exactly the same. Also, as a result of that, in the 20th century, if we did want to understand what happens below the wavelength and at the molecular level, we needed different tools. And that's what this community is about, right? What I call the three main pillars of 20th century biology: biochemistry, molecular biology, and structural biology. And they've been enormously powerful tools, but they have one common limitation, which is that they're all reductionist approaches about trying to look at the building blocks of cells rather than cells in their totality. And life is the most complicated machine in the universe. And if I want to understand a dynamic machine, no matter how good my tools are to look at the parts, I cannot reverse engineer a car engine from a random pile of engine parts. And that's kind of where we are. So we lose all the temporal information, we lose all the relationships. And boy, wouldn't it be great to be able to see what's happening at that molecular level directly. So the thing is because of those limitations, when people try to come up with models of what's happening dynamically in the cell, basically they hallucinate, they imagine what happens. And so these types of images and stuff, although they look beautiful, and although they do represent a lot of what we've learned, they're really at best caricatures of something much more complex and dynamic and difficult to understand that's happening inside of cells. And the reality is that starting from single molecules that are moving stochastically in a dense gel, they eventually start to aggregate and form macromolecular complexes that eventually grow into bigger structures such as organelles. Those organelles form units that are autonomous called cells that can reproduce. These cells then interact with one another, and then eventually they interact in large groups to create organs or us. And so life consists of many levels of emergence of different properties where complex systems come together to create something bigger. And stochasticity at one level leads to determinism when you have law of large numbers that's trying to cause things to happen. So fortunately, I came into the story with blind luck in the 1980s when various unrelated technological advances allowed optical microscopy to get out of its funk. The first, of course, was the transistor, which led to integrated circuits, which led to PCs that you could use to control microscopes. It also led to the development of sensors like like CCD chips that were much more sensitive and allowed us to create digital copies of images that we could then process and do things with. And so, for example, video microscopy came up in the 1980s, which was our first ability to take movies of live cells. Also in the 1980s and 1990s, this was the beginning of a revolution in chemical biology to create fluorescent probes that would be very specific to different proteins. So now we have protein-specific contrast of live cells that we can then see in our microscopes. And the final piece of the puzzle was in the 60s when the laser was developed. And almost every modern microscope that I'm talking about is all about taking that coherent light and those pure colors you get from a laser and being able to use those abilities to mold and shape the light to do different types of tricks to see living systems better. So my personal story with this began in graduate school when I went to Cornell, and there were two professors who had this crazy idea for what if we could look at living cells with the resolution of an electron microscope. And their idea was to take a hole which is much smaller than the wavelength of light, in this case at the tip of a optical fiber that we would pull, coat the outsides with metal, and then if the hole is smaller than the wavelength of light, if we put that right next to the surface, we have a little nano flashlight that we could scan point by point across the specimen and get an image better than the diffraction limit. Eventually, I took that to Bell Labs, where I worked for 6 years. At one time, we used that to have the highest data storage density in the world by writing bits optically instead of magnetically. Eventually, I was able to do super resolution for the first time. This is looking at the actin cytoskeleton in a fibroblast by normal microscopy and by near-field microscopy, as we called it. And eventually, I was able to show that the sensitivity of this microscope was such that you could even, for the first time, resolve single molecules. You wouldn't be able to resolve them densely, but if they're separated from one another enough, you could look at them and even determine the orientations of their molecular dipoles on a surface. So all those were great hits, but the problem was I lived that technique for 12 years. I was just completely obsessed with it, but eventually I found out that it had a dead end. And the dead end is the light that comes out of that subwavelength hole spreads extremely rapidly. And if your little aperture is not within 10 to 20 nanometers of the thing that you want to look at, the resolution very quickly degrades. Well, my whole dream again was look at living cells with the resolution of an electron microscope. Well, I didn't know much biology then, but I knew a cell was a lot rougher than 10 to 20 nanometers. And so this approach was a dead end for trying to be able to look at living things. So I quit my job, and I was done with academia at that point. I didn't know what I wanted to do. One day my wife still had a job, and we had a baby, and so I was pushing my daughter in a stroller, and it hit me that there might be a different way to beat the diffraction limit based on that single molecule observation. So the idea is that if you have fluorescent molecules that are dense enough to really see what you want to see in a cell, in a regular optical microscope, each one of those is a fuzzy ball. And the problem is all the fuzzy balls overlap and you get a smush. But let's say, for example, that all the molecules were in different colors. Well, then you could build a third dimension to the data set. And in that third dimension, each molecule that's a different color would be separated from one another. I can then point to the center of that ball for each one with much better precision than the diameter of the ball. And then, therefore, I localize or find the position to every molecule. And in that era, there was enough photons from every molecule, and detectors were good enough that you could localize it down to about 10 nanometers, even though the ball was about 300 nanometers. And so then if you project all of those locations back to the sample, you essentially would have a super resolution image. The problem was there was no good technology at the time to, by color, separate the hundreds of molecules that might be in one diffraction limited spot. And so we were still stuck. So then I went to work in industry for 8 to 10 years for my dad's company in Michigan making machine tools. I came up with a new machine tool architecture that failed spectacularly in the marketplace, and then I quit that job, and then I'm in my 40s and completely unemployed for the second time and don't know what I'm going to do. I reconnected with my friend from Bell Labs, Harold Hess, who had gone into industry when Bell had shrunk down. And so I had an idea for a massively multifocal microscope that I was trying to sell that I developed while unemployed. And we went to different places to see if I could find a lab where I could build it. One of the places was Florida State, where we learned about a new type of fluorescent protein. Normally, fluorescent proteins, you throw blue light on it and it glows green. With this one, nothing would happen unless you first put purple light on it to activate the molecules, and then the blue light would cause it to glow green. Well, Harold and I realized instantly when we heard about that, oh my God, if you turn that purple light down so low, only statistically a few molecules would come on at a time, and their fuzzy balls would be separated from one another, so we could find those centers, turn that subset off, turn on another subset, and go on and on and on, and then you get going from this to this. And so we said, oh my God, that's crazy simple. It's the idea I had 10 years ago, but now with time instead of color. Problem was is that he had quit his job, so now we're both unemployed. So the good news is that, well, two things. One is that he's a pack rat, so when he left Bell, I told them all to go to hell when I left, but when he left, he was able to take all of his equipment with him. And so we took that out of the storage shed and put about $50,000 each of our own savings into it. And we said, there's no time to start a company, there's no time to get a grant, we got to do this now because it's so easy, why hasn't anybody done it? And so normally you would do this kind of entrepreneurial thing in a garage, but Harold wasn't married, so we could do it on his living room floor. And within just a few months, we had this microscope. We made connections at NIH with Jennifer Lippincott-Schwartz, who was actually the inventor of these photoactivated fluorescent proteins. So we took our little microscope to her lab, and within just a month or two, we started getting images like this. This is looking at the membrane of a lysosome with the photoactivated molecules expressed inside of the membrane, and you can see the single molecules turning on one at a time. You end up, if you sum that up, you get the fuzzy image you would have in a diffraction limited microscope. But if instead you take each of these random images and plot the balls inside of their positions, you start to build a super resolution image. And you go from the one on the left for diffraction limit to the one on the right, and boom. So you can get a gain of at least 10, if not 20-fold, with a microscope that's simple enough you can build on your living room floor. So immediately this took off, and immediately it led to us getting jobs with HHMI and Janelia. So we both worked there, and it was sort of happy ever after from that point, except for one thing is because I'm always angry and unsatisfied with the tools I build, and Palm is just one more of those tools at which I get angry and dissatisfied. The problem is yes, we did a number of examples, but the problem is that it's so slow to bleed out all of those molecules one at a time that it takes a long time. And so largely we had to look at fixed structures, chemically fixed structures, to have the time to do that. That's not looking for living things, though. So the thing I learned in the end is there is no such thing as a free lunch. If I want to have ultra-high spatial resolution, that means my image has to have many pixels. If it has many pixels, it takes more time to make and measure those pixels. It also means I'm throwing more potentially damaging light at this specimen. And it takes more time to do all of that. And so there's always these trade-offs. I always say to my postdocs, if you don't understand the trade-offs, you don't understand the problem. Always trade-offs. And so the trade-off with Palm again was that you need many, many, many molecules to densely label the thing you want, to localize, to see the whole picture. And that takes a lot of time, usually too much time to look at a living specimen. But then once I was at Janelia, the head of HHMI at that time, Bob Tejan, had a lab there. And he's an old biochemist who has spent years unraveling the whole mechanism of transcription. And so in his and the field's work, the model that existed at the time was that many different transcription factors would have to come to the DNA, bind to one site to create a stable complex called a transcription initiation complex. And it was believed that this process would take hours to develop before recruited the polymerase and started the process of transcription. Tej wanted to see this in our Palm microscope, but not by seeing the whole thing at super high resolution like structural biology, but instead to see the kinetics of the single molecules and how they go around or bind to the DNA. When we did that, we found that none of the transcription factors bind for even more than a few seconds at a time. So this whole model that had been built up by biochemistry into how transcriptions happen was completely erroneous. Again, it's the hallucinations you do because you don't have complete information. What's actually happening is that many of these guys have intrinsically disordered regions on them, and so that they come together and they're sort of sticky near the site, and they sort of glom together, but different partners go away, different other ones come in, and eventually stochastically you recruit the polymerase and start transcription. So this idea of not trying to get spatial information with many, many molecules, but try to get kinetic information was for me revolutionary. It was like, oh my God, I'm not looking at anything dead ever again. Okay. And so it turns out that this is really important for different diseases where these intrinsically disordered regions, like in Ewing sarcoma, where a intrinsically disordered region is grafted by a translocation onto a different gene and then sort of causes an overexpression of certain genes that lead to this cancer. And so then Tej and I, plus a couple other partners, realized, hey, you know, maybe this single molecule screen to understand the kinetics might be a good way to do drug discovery. And so we founded a company in 2019 called Icon Therapeutics, which uses single molecule tracking to screen hundreds of thousands of compounds against particular diseases, mainly cancer right now, but some in neuroscience. So today, Icon is, you know, in Millbrae in California. They have 30 high-throughput microscopes doing single molecule tracking. They take 1 petabyte of data a day. That's 1 million gigabytes of data a day of single molecules tracking. And what's amazing to me is I didn't think this was going to, you know, I'm a founder, but come on, really? Okay, so anyway, so I went along for the ride. And they were able to hire the head of research from Merck, who came to be our CEO, and Roger Perlmutter, and he's amazing. But he's the guy who led the Keytruda project, which is, of course, their blockbuster cancer drug. And so one of the things that he decided is he wanted to understand all of the protein-protein interactions that he could never elucidate while he was at Merck. And so what he did is he created a whole genome, 1 gene at a time knockout screen of each of the target proteins we were interested in, and then look at how the kinetics change when you knock out 1 protein at a time. And by doing this, develop these huge trees of protein-protein interaction networks that were completely unknown before, and potentially lead to other secondary or tertiary targets for drugs that might do better and might be easier to develop than the actual primary molecule that you're trying to get, but you can change the primary's kinetics in a way that's useful. And so as you can see here, there's a clinical pipeline. The first molecules that they have are actually in-license from China, so that has nothing to do with single molecule screens. But our first phase 1 trial is ongoing now with the first compound coming out of that screen, and there's many more in the pipeline. So again, no such thing as a free lunch. So while the entire world in the lead up to the Nobel Prize was thinking, spatial resolution is the thing, and everybody is marching to that peak, if there's one thing that's consistent in my career, if a bunch of people are doing something, I run in the opposite direction. I don't want to be one of the crowd. I want to carve my own space. And so as I was thinking, is there a way to do something that would be informative in a different direction? So what did I learn from all of this? I learned that all biological and structure and physiology comes from dynamic processes occurring at spatial scales, from single molecules to whole organisms. When studying these processes, you have to have all the players present, or else you're looking at artificiality. You don't know what's going on. Optical microscopy's most unique and transformative aspect is we can look at living systems. And animation and natural variability are the fundamental hallmarks of life. How can you truly understand life if you don't look at it live? So that's been my mantra ever since. The problem is most of the time when we do live imaging, we're looking at cultured cells on a cover slip, which is pretty artificial, and we're looking at them usually in 2D because that's the way our brains work. We think in 2D, we image in 2D and time. Okay? But cells operate in 4-dimensional space-time. So this was an opportunity for us, and so it was like, instead of marching in the direction everybody else is, can we back off? Can we make a diffraction-limited microscope that would be as transformative for high-speed, non-invasive 3D imaging of cells as Palm has been for doing structural imaging high resolution? There was an opportunity here because at the time, the 2 most common microscopes are the 350-year-old wide field microscope, and as many of you probably know, this confocal microscope that scans a laser point in a cell, point by point, strips out the out of focus light the wide field doesn't, and gives a more clear image of what's going on. A super important innovation in microscopy, unrelated to me, that happened around 2000, was when several groups decided that instead of bringing the light from above and blasting the entire thickness of your specimen, bring a second lens from the side and create a sheet of light, which is only coincident with the one plane which is in focus at a time. That's called light sheet microscopy. If you then move that light sheet up and down, plane by plane, with the focus, you end up getting a 3D map of your specimen live. So that's been transformative for understanding embryogenesis at single cell resolution. But the problem is that because of, again, our good friend, the diffraction limit, there's a trade-off between how big of a specimen you can look at and how thick the light sheet has to be. And so at that time, there was a limitation to being thick enough to see a cell, but not much of what's going on inside a cell. Well, again, when I was unemployed for the first time before Palm, you remember I had some idea for a microscope that would use different types of tricks to do multifocal imaging. It turned out those same tricks, which involved what are called non-diffracting beams or Bessel beams, allowed us to make much thinner light sheets. And so what we could do is use my theories on this to make an array of such beams that would make an ultra-thin light sheet smaller than a cell, that we could drive the light sheet or the cell through it one by one and plane by plane build up a three-dimensional image. That became known as lattice light sheet microscopy. I was actually working on the proofs of this paper when I was in Germany and picked up the phone, and Stockholm was on the phone to say that I was going to get the Nobel Prize for Palm, and I was like, I don't give a crap. I mean, this is the thing I'm doing now. This is great, you know. And it was really disconcerting because I was so invested in this at the time, and I didn't need that distraction. But I have made many, many microscopes. I've done many things. This is the one I will take to my grave and love the most, because nowhere have we've learned more biology and had more fun. At Genili is perfect because you can bring in outside visitors. We had 100 groups come to our lab, and every one of them was studying the systems they've been looking at for 30 years and seeing them in a completely different way than they've ever been able to see them before. And they all go back with big smiles on their faces and 10 terabytes of data on a drive, and 2 weeks later they call us up crying because they have no idea what to do with 10 terabytes of data on a drive. You laugh, but that is the point of the whole rest of the talk, because it is so frustrating. This is the problem of where we're at right now, is that you can't deal with the complexity of the data. So there's yet another limitation, is again, I've been learning biology by osmosis because I'm not a biologist, but I did come to realize that cells on a glass slide are not exactly physiological. We know the phenotypes we observe are the result of gene expression. We know gene expression is controlled by the local environment. So if you're trying to look at cells in an artificial environment like a cover slip, how can you trust the results you see no matter how good your microscope is? You have to go like Willie Sutton. He robs banks because that's where the money is. We have to go into organisms to be able to study subcellular biology the way it really is. Look, a neuron is not like a kidney. They both have the same DNA. Why? Okay. The problem is that, again, we're stuck because cells have different refractive indices, so that when you look at something like a zebrafish embryo, your light gets scrambled going in, just as if you have water on your windshield before you turn on the wipers. And so you're stuck with getting good resolution. Well, astronomers figured out the solution to that problem about 50 years ago. It's something called adaptive optics. In their case, the atmosphere was screwing up their view of distant galaxies. You shoot a laser into the stratosphere to create an artificial star. You look at how that star has been aberrated and screwed up by the atmosphere. And then you figure out what the aberration is, and then your telescope has a mirror which can very rapidly change its shape to exactly cancel those aberrations. And you do that, and you go from this type of blurry thing to that. Well, we stole from them immediately. So this is turning on the adaptive optics, as you can see here, in a zebrafish embryo looking at neurons in the brain, and you can see how it goes from fuzzy to clear just by doing that technique. So eventually we combine the lattice light sheet with the adaptive optics, and then that allows us to now see what's happening in tissues. So here you're looking at a cancer cell that's starting to go through the process of extravasation, trying to get through a capillary. And so it sends out sticky integrins and then eventually starts to burst through the side and come out. Here you're going to see a T cell that's starting to move inside an environment here. And so this is in the ear of a zebrafish. This is the skin. This is the hindbrain. And this is a capillary here. And pretty soon you'll see this little guy. Well, there he is. He's coming in and out looking for things that he's supposed to eat. Okay. So basically, we're now seeing things as they really are in real specimens. Here you're looking at development, looking at axons growing inside of the spinal cord of a zebrafish, many, many things. So with the modern microscopes we have, from palm all the way up to lattice light sheet and adaptive optics, we cover an enormous range of space and time, 7 orders of magnitude time, 14 orders of magnitude of volume. So what does it mean, though, that if I have microscopes that have high resolution over large volumes of tissue with very high speed for very long periods of time? It means data, lots and lots of data, okay? So we spent a lot of time developing pipelines in order to be able to process this data and put it in forms that we can then see, okay? So we can take data at 4 terabytes per hour per camera in our microscopes. That immediately goes into a high-performance computing cluster, parallelized, does all the processing it, and you get pretty movies like this, or you end up getting, basically, this is an example of doing something called expansion microscopy, a different form of super resolution, looking at axons across the entire mouse olfactory bulb. Half a petabyte of data, so 500 terabytes in 10 days. But then the image, all the processing was done in real time. So this is great. So we have all of this, but even if we get to the point of rendering a movie, like here's a little chunk inside of a living zebrafish brain looking at 4 organelles at once, okay? There's no way you can understand that. Life really happens in 5D, 20,000 different proteins, X, Y, Z, T, all happening at once, and it's completely incomprehensible even after you take the data. We were evolved to see this guy, the lion, because we look in 2D plus time. So we're stuck. There's so much complexity that's going on, and we don't have any way to do it. So I was the last guy on earth who wanted to have anything to do with AI because, again, I don't like to go in the same direction that everybody's going, but God damn it, I'm forced to because there's just nothing else possible. We have to build minds that can see across vast 5D data and then report it back to us meatbags in the end in a way that we can understand. And so we have to be able to go from taking the data, having the computer automatically segment the cells, then automatically segment the organelles, do all of the tracking and all of the stuff which is laboriously done now by armies of graduate students and postdocs on a completely unscalable level. All right. So the roadblocks that we face now is the microscopes that are most revealing are too complex and expensive for individual labs. And trust me, I tried for 15 years to get our technologies. We did open source everything. I have a whole talk online on YouTube called Confessions of a Frustrated Optical Microscopist, in which I rant for an hour about why tech transfer doesn't work. Okay, but you can find that online. Second one is the image data is too vast and complex for the storage and compute resources that individual labs can have. And finally, the state-of-the-art AI vision models that exist for trying to crack this problem are way beyond the resources of individual labs. So, astronomy faced this same problem 100 years ago. So up until the end of the 19th century, individual people, either because they were just curious and could do it with their own budgets, they were independently wealthy, or they got philanthropy from kings or whatever, built successively bigger and bigger telescopes. Herschel was one of the greatest practitioners of this. He built his 40-foot telescope himself in his backyard, and he was a composer during the day, and during night he was an astronomer. And he revolutionized our understanding of astronomy. But by the end of the 19th century, it just wasn't possible to build bigger telescopes at this scale. So it ended up becoming a philanthropic effort or a government effort with bigger collections of resources and people. Hooker telescope, where they figured out that Cepheid variables to explain that the Andromeda Galaxy is 1 million light years away, that immediately exploded our concept of how big the universe was. And then the Hale telescope, which was the dominant telescope for nearly 50 years. So I'm a guy who is, my entire career, my group historically, up until about 5 years ago, was never bigger than 3 people. I don't like to have a big group. I like to be personally involved in the research I do. I consider every project a collaboration between me and a postdoc. But I can't do that anymore for this. So because of the problems of these resources, I feel that microscopy now has to, once again, follow astronomy in their model and try to build a centralized facility to crack this problem of being able to understand life as it is at scale. And so my best friend and longtime colleague, Gokul Upadhyayula, and I have started this initiative we call the Cell Observatory. It's just getting started, but our idea is just like in astronomy, biologists would come to the observatory with proposals of things that they want to see or things they want to understand. They would either have the reagents or we would build the reagents for them, the organisms and so forth. We have the microscopes, we take the data, we eventually put it into an AI model that we develop to allow them to then extract information that comes out of this. And it would all be open source throughout the whole thing, so all of the data would be available to everybody, the results from the models and so forth. So the workflow is people come with a whole series of questions from molecules to organisms. If the model has already been trained with the thing that they are asking about, cool. We don't need to do anything except query the model through a large language model to ask, hey, what happens when a T cell is trying to extravasate through a blood vessel? Show me 15 examples in your database. Show me what happens when this pharmacological perturbation that you had done had happened and how that affected it, and so forth. So we're going to build this massive database of what's going on. And then we get these eventually human interpretable results. So that's the vision. But, come on, okay. There's 4 pieces to building that vision. Oops, come back. There's first the reagents, and so we have to make decisions about what we're going to initially train the model on. I'll get to that in a minute. Then they have to all be imaged. That's the thing we're good at. We're really good at that. And the next thing is data handling. We're pretty good at that. Then there's the AI, and we are neophytes at that, but we're living in the right part of the country to try to find people who are really good at that and trying to work with them. So we've decided initially, I don't know what's wrong with that one movie, but start with zebrafish as a model organism. We like the fact that it's a vertebrate, has 60% homology in its genes to humans, and so we're starting with that, but that certainly wouldn't be the end. So all of a sudden, instead of 3 people, now Gokul and I are up to like 20 people. So the first team is our zebrafish team, who's developing first the plasmids, the RNAs, then we have transgenic fish we're developing, gene-edited, and multicolor fish for segmentation we call Skittles. So you go from easy and quick stuff to, I love fish. It's for a microscopist, it's God's gift to us that there's this nice transparent vertebrate we can look at. I hate the fact that it has a 3-month sexual maturation time because, man, this pipeline takes a long time. So there's a ton of work that goes into this to use state-of-the-art fluorescent proteins, to use state-of-the-art gene expression methods that do the best we can expression but still be physiological, and benchmarks to confirm that they're physiological. We have to, again, the other problem with fish is the transgenics and the knock-ins are nowhere near as developed the technology as it is for, say, worms. And so we're right on the bleeding edge of trying to do that as well. As you can see, we have some successes. So this is looking at dynamics inside of the fish with some of our transgenics that we've developed. Let's see. Okay, and you can see we got a big, long list of things. 20,000 different proteins, folks. We're not going to be able to see them all, but we try to hit the high points. So all the blue is things that are going to be done. All the little circles represent whether we've created the plasmids, the mRNAs, done any knock-ins and so forth first. The green ones are ones we've not only made but actually imaged and confirmed that they're good candidates. Ultimately, we're going to do about 500 ourselves and hope that we can convince other labs to start doing their own as well. Each one of these will probably require about 8 terabytes of data to fine-tune the model on. So then we get to the AI. So first we have to decide, biology dictates what organism we pick, what type of knock-ins, what kind of things we want to do. In order to understand what kind of microscopes we need, what kind of compute we need, and so forth, we need to get our feet wet with understanding the AI models themselves. So we started doing that. So these vision transformers exist. They largely developed for autonomous driving. So they learn spatial-temporal relationships across all scales in their data set, which is exactly what you need in biology, because everything is happening from molecules to the whole thing. It requires, the nice thing about these transformer models is you don't need to have a human has gone through and annotated and said, every bit of these images is this, this, and this. It can work on unannotated, pre-trained data first, but vast amounts of it, and then a much, much smaller subset of data that's been annotated. Fluorescence annotates things itself without humans. I can put in a marker that says this is this cell type or that cell type. The model can then do that. We need to reduce the amount of human content that goes into this whole process. So to learn the scaling for this, we try to do an experiment where, remember I said we do adaptive optics. First step of adaptive optics is to learn what the aberration is. So we've figured, well, we'll see if we can get an AI to just look at the images and know what the aberration is. And we succeeded in doing that. And so from that and doing what's called ablation experiments where you figure out, well, did I make the model too big or is it not big enough? And what happens if I do this, this, and this? You start to get a sense of how things scale if you want to do these types of vision transformers. And what we find out is that, you know, what we want to do in terms of creating not just a 3-dimensional model there, but again, a 4 or 5-dimensional model for a vision transformer, it's potentially doable. It's right at the limits of what the largest large language models on Earth are now, like from Google. It's about at that scale, but our training experiments suggest that we're close in terms of the size. Our estimates are probably accurate of what would be involved, but it's big. It's bigger than we are, and ultimately, if we don't capture the interest of a Google or a Meta or something like that, this thing will probably die. But we're trying to skate to where the puck will be in terms of this acceleration of AI resources and so forth. The other problem when you talk to those companies that they say is they have great compute, they have great people, but they've scraped everything off the web long since and they don't have enough data. Okay, they talk about peak data like they used to talk about peak oil, right, that we're running out. We will fix that problem for them. I think this is a marriage made in heaven because nobody produces better quality, quantity data than we do. So I'm keeping my fingers crossed. So how much 45 data is enough? Anywhere from half a petabyte to 50 petabytes. So what that means is we need a lot of data. And our microscopes are performative, but they're not necessarily that performative. So the microscopes we do have and that we've been using, and almost all the images I've shown so far, all these movies, have come from something we call Mosaic, which uses adaptive optics and lattice light sheet. And it gives us really great views like this. This is inside a zebrafish. This is the spinal cord region in the middle. This is the lateral line primordium that's moving along at the surface. This is, again, in the middle of the spinal cord here. Axons growing here. So it's a beautiful, I mean, I'm blessed to have been a microscopist because I've just fallen in love with the beauty and complexity of life. And I feel like I've many times felt like Galileo when he's first pointing his little 30x telescope at the stars and seeing things nobody's seen. I feel like I've seen more of life in the way life really is than any human on Earth or in history. And it's a real privilege to be able to see this beauty everywhere. The problem with this microscope is the way it works. You can't really easily put chemical perturbations on it, and it's a bitch to put in one fish and take out another, so it's slow. So we need more microscopes, better performative microscopes. And if you see our performance so far in terms of what we need for the model, in terms of getting the pre-training data for segmentation, we're not doing too bad, but for everything else, we're about 10% of our goal with those two microscopes running for a year 24/7. So we need more microscopes. Well, you remember I talked about ICON and that they take a petabyte of data a day. Well, the way they were able to do that is when the company started, I designed a microscope for them, which uses a tilted light sheet that just sweeps very rapidly across the specimen, only takes data from the sweet spot of the light sheet that's in focus at the focal plane, and very quickly then does single cells. So I didn't think this microscope, because it sends so much light to the deeper parts of the specimen, even though you don't directly look at them, I didn't think this would be that performative for doing multicellular imaging. But Kokul said, I had a postdoc, Amir, who was doing single molecule tracking to study epigenetics, and said, well, why don't we use that microscope and throw an embryo on and see what happens? And what happened was this, and I get, oh my God, I didn't expect that. So it's really quite, this is looking at the ear of a zebrafish. You're looking at the lens of the eye with all those thin cells there and these other cells around it. This little bub happening is erythrocytes moving in a blood vessel. This is life. This is what's happening. This is a tiny, tiny bit of the complexity that's going on. But that microscope worked great. So we've developed a new microscope we call ISOR, and this is now hot off the press. The microscope is just coming online. So this is looking at 3 different proteins across the fish at a resolution of about a couple hundred nanometers, or right to the diffraction limit. The green is, you're seeing muscle cells there. The green is membrane. The magenta is the nuclei going up to the eye here, and we'll zoom in, and these are mitochondria in each of the cells as we go deep. So seeing 3 out of the 20,000, okay? But this gives you an idea of how complex a machine living matter really is. And so we got 2 of those microscopes we're developing, and we're using it also to test the reagents of our pipeline as it's coming across. So this is looking at several different subcellular organelles. This one in the middle is the heart, and you can see actin there. The resolution stinks because, again, this is just hot off the press. This is just the imaging itself. We still don't have the adaptive optics functional on this yet. And we're also going to have a form of super resolution added to it called structured illumination that will double the resolution beyond what we have here. So this will be amazing. I mean, this microscope will be absolutely amazing. The Cell Observatory is a vision language model that we want to develop to unravel life from the single molecule level to the organelles to individual cells and finally to tissues. And so this is a big adventure for us. This venture's been going on for about a year now. We're, like all of you, struggling for money. And for me, I take a different model than Kevin. I've never written a grant in my life. Seriously. I've always lived off of philanthropy, whether it's been Bell Labs or Janelia, HHMI, or other funders. I understand it's the way government is the dominant source. It's the way it has to be. But I find that not having peer review, not having grant writing, it allows you so much more freedom to be able to and I've been blessed to be able to do that. And we're taking that same approach. And so, again, I'm going to have to leave this meeting early because I have to try to chat up a potential donor at dinner tonight and see how that goes. But anyway, that's where we're headed. It's been a really fantastic ride so far. And I'm really, really more excited than ever to see if we can get over this hump of the AI, how transformative. I don't think you I can't, maybe I'm being grandiose, but this is going to be so transformative that it will completely change the way cell and developmental biology is done. There will no longer be this sort of a grad student at a bench doing a knock-in, doing, oh, I'm looking at mito-ER contacts, right? And I do a thesis on this, and I have a thesis, and I I take gigabytes of data, which is distilled into 2,500 words into some magazine that's behind some goddamn paywall with 6 figures, and that's it. And then most people are never even aware of it in the end, right? Even if it's good results, the scientific enterprise is so vast, you don't have... We need to build models that are inclusive and can guide us through what the complexity and size of science as it is. And the cell observatory, as part of that model, will revolutionize the way biology is done. You will be able to do theses by querying the model, and if it's not in the model, you're going to build the reagents and so forth so the observatory can add it to the model, and then you will mine that data through the model to get your results. And the results you'll have will be digital and available to everybody, not through some damn paywall. That's the vision that I see. And if it gets agenic, if agenic AI happens, the models will be your thesis advisors. They'll be the ones who are guiding what you're doing and what you're learning. So that can be dystopian too, I understand, but there's many dystopian aspects of it. But that is a possible future that might happen within a decade. So anyway, leave you with that sobering thought. Anyway, thank you very much.